you are saving on your computer, like a TXT file or like some type of a CSV file that has the genes type, genes with the adjusted p-value and then log to file change, something like that. You have it now? If you don't have, I'll just run that code maybe one more time, and then we can go through that one more time, and then we'll do the some functional enrichment analysis of those differentially expressed genes that we have. But we might be meeting again on Thursday, so when we meet on Thursday, I'll try to put some slides where we'll go on the basics of some of the program, like how DESeq exactly works, like step-by-step process, and then how PCA exactly does the dimensionality reduction, how it is doing those visualization, and then what is the normalization in RNA-seq, what is RPKM, what is FPKM, what is TPM, and how those are different from the normalization that DESeq to ASR uses. So we can discuss this basic stuff as well, and then create some more other fancy figure like heat map or like some other figures on first class. So just to go over this overview again, so we have, when you get the sequencing results, you get millions of reads as a short sequences from the sequencer. You need to do the quality check, and then after you do the quality check, you will have to align those short reads against your reference, either genome or the transcriptome or any different gene models that you have that you are interested to quantify the expression of those genes, right? And then after that, you do compare, use some statistical model to identify the genes that are differentially expressed between the control and then your interest of treatment. And then do some functional enrichment analysis and other downstream pathway analysis that you are interested in. So we created, as a first step, as we talked last week, we created some principal component analysis, which is one of the dimensionality reduction approach, where you can visualize the sample, how those sample or the biological replicates, or between the sample, how the transcriptome, or if you have the single cell RNA-seq, your single cell RNA-seq, or if you have the metabolomics, or you have the proteomics or lipidomics data, how those samples vary from one to another one. Basically, it helps to reduce the dimensionality from all those 27,000 genes putting in one dot and then trying to visualize how that is different from other samples, right? So this is one of the examples for principal component analysis, where you can see the control sample here means it has the three biological replicate for control, and then same thing for other salt treatment, 3 hour, 24 hours, 72 hour after salt treatment. And then you can see the these transcriptomes of each one of these samples are distinct from one another. That's what we can say from here, right? So control sample has a distinct transcriptomes from 3 hour, 24 or 72, and each one of those samples has a different with each other. That's what it means here. And then if you see here between the species, if you have a multiple species, you can do that for multiple species as well. And then if you are comparing human different genotypes, different sample of human, or different plants like rice, maize, or other different crops, then you can put the ortholog genes, the similar gene, most similar gene between the species, and then create those and create the principal component analysis here. If we are talking about the two species here, you can see the biggest difference here is coming between the species. So these dots on the left are for the one species Arabidopsis, and the right is for the Fabula, or the other species. So you can see the biggest difference, 88% of the variance is contributed by the species difference here, right? And then if we see the only one species, then you can see the treatment effect here. So you can create or you can visualize this type of data set and then see how the biological replicates are behaving. So if you are dealing with the field sample in most of the ecology or like other experiments, if you are collecting sample from field, at that time it's very difficult to have the samples that are agreeing or those has the similar transcriptomic profiles with each other. So having like multiple replicates will help to do your analysis. So anyway, most of the journals nowadays require at least 3 biological replicates. So when you design RNA-seq dataset or RNA-seq experiment, like it's almost impossible to publish when you have less than 3 biological replicates, unless you have so many other dataset like physiological dataset or other type of validation that you have, and then you can just to see some interesting gene like 1 or 2 gene expression pattern, then you can use the 1 replicate or 2 replicate. But even when you have the 1 biological replicate, at that time if you use some of the statistical tool that can simulate the other replicate, so you need to use the right tool at that time. So the tool that we are using in the class, so that's DE-seq tool, right, to identify the differentially expressed genes. So when you use that, it doesn't simulate the replicates. So you don't have to do that. So and then you, if you did the DE-seq tool over the weekend, so you will see this type of a plot. So where you will see so many gray dots and few either red or blue dots, you can manipulate this plot, make it as pretty as you want. And then, so whatever this red dot above this 0 line here, this is the log2 fold change, where log2 fold change is calculated based on the expression of that, each dot here is the gene. So for that gene, the salt, expression of that gene under salt versus the control condition. And then if the fold change is positive, this is in the log again. That's why positive means the higher expression upon salt. And then when you have the negative log, that means down-regulated. So this many number of genes are down-regulated, and this many number of genes, all these red dots are up-regulated upon salt stress here. That's what it means. But most of the time, your RNA-seq data set, when you design the experiment, you won't just have one-to-one. So you will have a multiple different either time point or different sample or different developmental stages, right? So you will have to have a lot of pairwise comparison. If not, you are doing clustering. So if there are like 100 sample of RNA-seq from like 13, 14 different developmental stages, then it's too much to do this type of differential gene expression analysis. Rather, you will be maybe interested doing some type of clustering analysis where you will see the pattern of gene expression rather than what are the genes that are up or down. You may be mostly interested in the pattern. Okay, with the developmental stages, these are the genes that are going up, or these are the genes that are going down, or these are the genes that never change, doesn't matter what developmental stages. So it just depends on what type of RNA-seq experimental design you have. So you choose the tool afterward. So but if you haven't run the DE-seq tool, we can do it today later. But I put 1 or 2 more slides just to give you a broad sense of idea for the experimental design in RNA-seq. So most of the time we will be confused like whether we should sequence more, like more sample, like you put because how the sequencing machine works is you have this flow cell, right? So and then in this flow cell, each flow cell here, maybe Illumina has 8 of those, and then it depends how many. For example, this one, if you spend the entire flow cell, you will get 100 million reads. And then if you have prepared the library with like 10 different samples and you put in one flow cell, basically that means you will have per sample just 10 million reads, right? If you want to spend all the flow cell for 10 samples, 1 sample, or you want to include the biological replicate. So which one makes more sense for you? So that type of decision you have to make it when you are, that's the first decision actually you have to make when you are designing a RNA-seq experiment. So this actually helped myself when I was designing my experiment in 2015. So so each one of these is number of reads. As I told you earlier, when we have the sequencing reads comes from the machine, you will get it in millions. Okay. So that one is, if you see the number of reads here, it's in millions, so 5, 10, 15, 20 millions, and this is the number of replicate here. If you have 10 million, and this is the number of differentially expressed genes. So basically, with increasing the biological replicate, your chances of getting the more number of differentially expressed genes is higher than just having 2 replicate or 3 replicate. And then if you see the, when you increase the number of read, the number of differentially expressed genes that you are getting doesn't increase. But when you increase the biological replicate, then the chances of getting more DEGs or getting the capturing the more genes is increasing significantly here. That's what it is saying. So sequencing depth here means how many reads you are trying to get. How many of these flow cells you are trying to use, or how many of these lane, in Illumina you call it lane, but in PacBio we call it a flow cell. So how many lane you want to use it for your sample. Based on how many lane you used, that will determine how many number of read you are getting. So those are the reads ultimately going to map with your genome here, right? So if you have 100 million reads, where it will match. Or if it has the 10 million read, so it has the inherent bias for the genes, right? So how many times the reads are mapping to that gene. If you are sequencing 100 million reads versus 10 million reads, definitely there is a bias with the expression now. So you want to, that's why this is very important step, how great you want to sequence your sample, or how many biological replicate you want to sequence. At least you will always have to have at least 3 biological replicates. And then it's kind of impossible to harvest all the sample at the same time and then extract all those RNA at the same time, right? Same day. You are processing all your 50 RNA sample today. No, you can't extract all those at once, right? And then you can't harvest all those at once. So there will be always batch effects there. So you have to also account for those batch effects when you analyze the RNA-seq data. So if you read the D60 manual, then you will see a way to do the batch correction. But that step you will decide only after seeing this principal component analysis or something other method that does the this dimensionality reduction, like t-SNE or UMAP. So those are basically being same things here. So if this biological replicate doesn't agree with each other, for example, one of these, if it goes and stay here, so and then 2 of the replicates are here. So basically now you have to find those solutions. What you're gonna do that with that sample, right? You can't remove that, but you have to find a way to somehow, like you will assume you might have introduced some unexpected variable when you extract RNA or harvest sample or prepare library or sequence. So you're trying to basically correct for those batch effects. So that one you can also do it in DSeq2 and then also in NuiSeq. So those are basically 2 different types of, totally different type of programs that can handle RNA-seq data. We'll talk in a little bit. Yes.
I have a question. Can you please go back? What's the difference between the 2 plots, upper and down plot?
Or here and here. So this is 2 different tissue types here. So this is for the root and this is for the shoot. And then root and shoot harvested from the same plant. And then control 3 hour 24/7 after the salt treatment. Yeah. And then this is for the tree species. So for the ortholog gene pair, if you are not familiar with ortholog gene pair, maybe we can talk about it later, but each species has their own set of protein coding genes, right? For example, maize has like 20,000 genes, and then Arabidopsis has 27,000 genes, right? So when you are comparing the 2 species at the same framework here, then you will only can compare those with the same set of genes, right? And one is 20, one is 27. You cannot compare those. So it's like comparing the apple and orange. So you can compare just apple versus apple, right? So at that time you are trying to calculate the most similar gene from Arabidopsis. In this case, from Arabidopsis 27,000 gene versus the other species SP, and then it has also like 27,000 gene. But these two species are from the same family at least. So you need to find the, for example, this gene 1, gene 2, and then gene 3. What is the most similar, most closely best hit, most similar gene from the Arabidopsis to this species here? For example, this is gene I, and then both, and the gene I from this species also finding the best hit in Arabidopsis, and this gene 1 finding the best hit, most similar gene in the other genome. So that's what there were the list of genes, like out of 27,000, there were like 23,000 genes that can find the best hit from one to another. Those are reciprocal blast hit. They find the best match from one genome to another. So you put those ortholog pairs. So basically ortholog pairs, and then you now have the gene by sample matrix again. Instead of 27,000, now you have that table for 23,000 and all the samples from both species, right? And then you create the PCA for that. That's what the right figure is. Those are basically showing the two different things here, although they look similar. So the key thing is adding the biological replicates always helps to increase the power of your statistical analysis in RNA-seq data rather than increasing the sequencing depth. But this is solely for the purpose of differential gene expression analysis. If your goal is to identify the splice variants, for example, if you have the paired in sequencing and you are interested to identify the alternative splicing form. For example, this gene 1, this gene 1, so when the mRNA is formed and then processed, so this gene 1 can be processed into the different length, right? Gene 1.1, 1.2, or gene 1.3, or sometimes they don't go any splicing and just one copy is there for each gene, right? So if you are interested in this type of alternative splicing form, maybe going for the larger or the bigger sequencing depth is better. But just for the purpose of the differential gene expression analysis, more biological replicates is always better. So some tools, although in last class I didn't know that I can teach one more class, so I added a few slides here so you have some general idea. So we use, because this is like a gold standard for the general RNA-seq analysis, DESeq2. But you don't have to assume this is the only one tool that is available in market, right? So there are other tools as well. And then some of these tools basically uses the similar, they assume the similar distribution pattern of those genes or the gene expression value here. So one of the things that you need to consider is there are two different types of a method. One is the parametric method, one is the non-parametric. So non-parametric basically doesn't assume any distribution pattern there in the data set. But all these parametric methods, they assume some type of a distribution of those gene expression values. And then when you use the analysis tool, as last time we used the DESeq tool, but there is a non-parametric way. This is called the NOISeq tool. So where most of the time the suggestion for the RNA-seq analysis will be you choose the tool that assumes there is some type of distribution and there is no distribution in the data set. Like you use two types of tools. Basically you use DESeq tool and then NOISeq, and then you get the overlapping set of the differentially expressed gene. Most of the time this is like a huge overlap will be here. So if you have a DESeq tool and then you have the non-parametric method, this is a NOISeq, and then you get the conserved set of differentially expressed genes. Those are like high confidence set of differentially expressed genes. Basically you're trying to validate from two independent statistical approach here. So that's why it's always better to use more than one tool, like use DESeq2 of course, and then use NoisQual. And then since those two are the assuming totally different type of distribution, one assumes distribution, one never assumes there is a distribution in the data set. And then you get the conserved set of differentially expressed genes.
I have a question. When would you want to assume the distribution?
So most of the time, so when you have these RNA-seq data, we will see when you plot your data set, you will see some type of a distribution. But most of the time, these are like a negative binomial distribution that the regular distribution that we are assuming. But when you have your sample that is like one replicate, for example, where you cannot see any, like you cannot compare from the between the replicate, that time you assume there is no distribution there. And then at that time you have to decide, okay, I want to use the NoisSeq now. And then NoisSeq will simulate the existing data set there. So if you tell NoisSeq, okay, but you have to figure it out how to use that. So it uses some type of a machine learning algorithm. And then it will help you to simulate the other two biological replicates from the existing one biological replicate. And then it will use those final three biological replicate to simulate it one layer, and then calculates the significance there again. But when you see the DESeq2 one, so DESeq2 basically it computes the basically it has the this gene ratio there. So for each gene, when you have this type of matrix, so gene 1, gene 2, and then you have a gene 1, and then you have a sample 1, and then you have a sample 1. So this type of matrix, I by J matrix, and then you have some type of a value here, 1, 2, and then 100, and then it has some type of expression value. So it will first, everything in the DESeq2 is basically log. First thing it does is it will convert all these values into log value here. And then whenever there is a expression of 0, means there is no expression, then those genes in any of the sample here, if one of these sample has the expression 0, then this gene will be removed from the analysis in DESeq2. So the last week when I was talking about the sequencing, the library composition, that's the composition here. So, and then the sequencing depth. Depth is how deep you sequence based on here. But this type of 0 or no expressed genes will be removed from the analysis, and it will calculate basically the geometric mean here. It will first calculate the median value here, median value for all these values, the gene expression for this gene 1, calculate the median, and then subtract that, for example, this value here, x, minus the median value here, the median value, and then divides that with the standard deviation there. And then it will find some type of factor, and then with that, that factor is basically a number here. So that number, then it will be used to normalize each gene here. Basically, DESeq2 normalizes each gene there per sample. So this gene is normalized across the sample. The second gene is normalized across all the samples, and so on and so forth. So this is really the different type of normalization that DESeq2 offers. So if you understand it properly, then you will see why people are using DESeq2 there. There are a few videos, or you can just read the manual where it is explained really nicely there. But the key point is using the two different tools and then going for higher biological replicates over the sequencing there. So we talked about DESeq2, and then these are the input that we used last week, right? And then in the DESeq2, at the very last step, when we did the statistical test, there was a FDR, right, for false discovery rate. And then basically that is from the p-value. If you see the output from DESeq2, the table, either text file or the CSV file, there is one p-value column, and then another one is the adjusted p-value, something like that column will be there. So basically those p-values are adjusted to account for some of the false positive. Like if you are familiar with this type of type 1 or type 2 error, basically we are trying to account for this type 1 error here. So just to make sure you understand what is this type 1 error, so this is, I think, more easy to remember. So you can just read it. So it's a false positive result. So you don't want to miss those type of obvious DEGs from your analysis. So you want to keep those. So that's why DESeq2 is offering the false discovery rate correction for those p-values, those initial p-values. So how do you calculate the FDR is, let's actually discuss about this. So when you have like 20,000 genes in total in one organism, for example, so simply when you are talking about, okay, this is significant just based on the p-value cutoff of 0.05, right? So what does that mean is with the 0.05, there is a possibility 0.05 of those genes will be called as a differentially expressed by chance, right? That's what it is, the meaning of p-value, right? So that means out of 20,000 genes in general, this is the average number of protein coding genes in one organism, right? 1,000 of the genes will be anyway called by any statistical tool, okay, these are the genes that are differentially expressed, doesn't matter what. You don't have to have any treatment. That's what the p-value 0.05 means, right? Cutoff. So that means if you get 3,000 differentially expressed genes in any of your RNA-seq data analysis, that means, and 1,000 is already you will get it by random chance, right? Means if you have 3,000, then 33% of your genes are basically called as differentially expressed just by random chance. That's what it means, right? So we try to use this FDR concept to account for the type 1 statistical error that we are talking. So I actually found in my note how I was preparing for my general exam back then. So I thought this would be easier than just talking. So if you have the genes, and then each gene has the p-value there, right? So p-value, for example, these are the p-values for the genes, and then you rank those from ascending to descending order here, like, and then rank those 1, 2, 3, 4, 5, 6, 7, 8 for the p-values. And then now you are trying to calculate the FDR. So total number of p-values you have is, for example, if you have 27,000 gene, then you will have 27,000. Right now this is just example, so we have 8. And then ranking for the current p-value, like you are trying to calculate the FDR value for the 8th one. So the ranking for this is 8, and then what is the current p-value here is 0.75. So basically now you are trying to calculate you are trying to adjust the p-value, and then this value will be always smaller than what the value here is, right? So adjusted p-value will be not smaller, sorry. It will be the larger than the actual p-value is. So that's why you have to always adjust those p-values. If you see for this one 0.01, so this is the total number of p-value, and then you have the current ranking is number 1, and then the current p-value is 0.01. So you can see this will be non-significant if you have the cutoff of 0.01 here for differential expression analysis, right? Because the p-value is now bigger, 0.08. So it won't be called as differentially expressed genes anymore. So we are trying to impose some type of false discovery rate correction there. And then when you are reading the manual for most of this tool, you will have to figure it out what type of the correction the tool is using, or is there anything that the tool is going to use. So it's always the critical thing that you have to correct the p-value when you are doing this large set of differential gene expression analysis. It's just going like a Tukey test where you are doing multiple variable comparison, right? So you are trying to correct the p-values there. Okay, this one will come in a bit. This is another concept for the hypergeometric test where you are trying to see what type of functions are increased from your set of genes that you are using. But before we go here, maybe if you haven't had a time to run DEC2. I'll just show one more time, and then we can maybe move from there. So, okay. So I'll just go through the lines that we did last time. So we made the PCA. Again, when you like making the final set of figure for your manuscript for the that involves some RNA-seq dataset. Just there are a lot of resources available nowadays online, right? So just try to use those resources, and then to make your figure like really good quality figures. So having a good quality figure always give a good impression to the reviewer. If you are serving as a reviewer, and then you always want good thing, right, to see the paper. So just tweaking some code helps to make beautiful figure, and that helps to make your life easier in the publication process. So and then if you just Google PCA, DEC2, and then you'll see a lot of resources there, and even the general the MAplot thing that we will be generating soon. You can make it like so fancy. It looks like really good when you first see that. Like just have that type of I don't know, mentality or something. Go from here. So okay, we did calculate some type of. Okay, we were here. Did we calculate the some normalization factor, size factor? Okay, we calculated already. Okay, here. This is the thing that we were talking the other time here. So this value here. So when it is normalizing for each gene, and then at last it will have some type of a value here. So this is the value for each sample, because it is calculating, it did the all those normalization in the background and generated some type of a value for each sample there. And then now this value will be used to divide the initial raw count value of the gene expression. How to calculate size factor in DEC2? If you Google that, you will see all this process that I was describing here. But it's very clear in the manual. So we calculated the size factor, and then we did the PCA, but our job is to see the differential gene expression analysis. So this function here, results function in the DEC2, basically it uses this contrast function where you can, results function call for the contrast, and then it will basically compare the two samples. You asked for the sample comparison. Sample comparison basically coming from your this experimental design file, and then you ask to compare the this 24-hour sample with the control sample here. That's what it did here. So if you just run this code here, so it will calculate now, and then you can now just see. If you want to just see how the this object, what it is storing here under this, you can just see. So it has the some type of a table here, it seems. So where the first column is gene, and then it has some expression value, that's the base mean expression, and it has the log to fold change, it seems. And then log to fold change, it has some standard error there, and then it calculated the p-value, and then it has the p-adjusted value. Basically, these values are always bigger than this value here, because this is the adjusted value now, and it was adjusted based on what we earlier talked about. It adjusted the each p-value for from this table. First, it actually sorted, and then it ranked those p-values, and then it actually corrected the p-value there. And this is what we use. We are going to use to say, okay, this is the cutoff I want for my differential gene expression analysis based on the FDR.
So can you repeat that? So this is the cutoff. When do you decide what the cutoff is?
Right now we haven't decided any cutoff. So in the following few command, maybe it will be there here. So I asked it to consider the adjusted p-value less than 0.01 as a DEG there. So if I ask that, then it will say what is the number of genes that passing this threshold. Since this is really a high salt concentration, and this species is a salt sensitive species, so many genes already responded to the salt stress here. So 10,000 genes are basically differentially expressed upon salt stress, or either up or down regulated. But you can always set up different threshold, like this is really stringent adjusted p-value cutoff. You can put the log 2 fold change. Some people also put the log 2 fold change cutoff. If the log 2 fold change is greater than 1, then only consider the differentially expressed genes. Right now I didn't put, I usually do not put any expression threshold there. Doesn't matter if it is lowly expressed or highly expressed gene. If it passes the p-value threshold, I always consider those as but it depends what you are trying to get. So there are some code here I put that you can still account for the this p-value, and then also the log 2 fold change. And then if the p-value is less than 0.01, and then the log 2 fold change is, you see here. So I didn't put anything cutoff here. If you say greater than 1, then you will get a less number of differentially expressed genes. But basically here I am trying to see how many genes are upregulated here. But if you want to get the downregulated one, you just have to change this to the less than 0 fold change, like negative values in a fold change. So you will get the downregulated genes there. So now you want to draw the figure and then see how it look. First I put one, I created one object here where I want to put the line, draw the line in the figure that I am going to create at -2 and plus 2. Basically I want to have some type of line followed with the like a blue line there, with this much of the thickness of that line. So and then this is the plot MA function. In it comes with a DSeq to itself. So you just use that plot MA function and then visualize the object that you created earlier with some alpha value for the dots there, and then you want to write the title for this figure, and then you want to give the y-axis minimum and maximum value -6 and +6, and then you want to draw the line. This draw line function object was created earlier here. So you can just plot it here and then see if it created anything. It should create, but maybe I couldn't see what it is. It's hidden somewhere underneath here. So yeah. And then now when you see this one, so in the x-axis basically these are the normalized expression. This is the expression value here, but in a normalized way. These are not the raw count value here. These are normalized based on the DSeq protocol here. And then y-axis. So because we asked in this draw line command here to make the to draw the line, dark blue line with this thickness by -2 and +2 here. That's why you drew the line here. You can make any cutoff here. So it will show you basically what are the genes that are like with a higher fold change that are differentially expressed, either upregulated or downregulated genes. And then you can change the color here with a few addition of the code. You want to change the dot size, or you want to make this upregulated gene as a different color, and then downregulated gene as a separate one. And you can also write the name here. What are the genes, either like really upregulated, highly upregulated, or highly downregulated gene. You can have